Hello, and welcome to the Alzheimer.tv News Bulletin, an initiative of the MoCA Clinic and Institute. This week, our team takes a closer look at a new blood test that may democratize Alzheimer's diagnostic. We review an amyloid-targeted treatment that showed positive results in its phase 2 testing, and we'll present the Biomarkers exhibit from our Alzheimer's investigation. The American biotechnology firm Amaranthus is currently testing a new blood test that may prove successful in diagnosing Alzheimer's disease. Kaya, you have more on this?
Yes, Eric. The lymphocyte proliferation test, known as Lympro, will allow physicians to differentiate Alzheimer's from other types of dementia with a simple blood test. The Lympro test assesses the proliferation of lymphocytes in blood associated to Alzheimer's disease. The Lympro test was first invented in 2001, but it was licensed by Amaranthus in 2012. It's now in its last trials before becoming available to the public, which could be as soon as the end of this year or early next year, according to Amaranthus. This test responds to a crucial demand in the field. Right now, the only way to definitively diagnose Alzheimer's is during an autopsy. PET scans are fairly accurate, but are not widely available to the public and not reimbursable in every country. Clinicians' diagnoses using the usual pen and paper tests will only accurately recognize AD approximately 70% of the time, according to Gerald E. Commission.
I think if we can get something into the mid-80s, I think that would be a home run for the field. I think that would be a grand slam. If you can get something in the mid-80s that has moved the sensitivity and specificity of the clinician's diagnosis towards the sensitivity and specificity of a clinician's diagnosis plus PET, and you're doing this in blood, we think, and I think everybody in the field believes that would be a home run.
At its release, Lympro would probably be used as a diagnostic help in combination with a clinician's diagnosis and followed by a PET scan. But the objective is certainly for it to one day become a standalone test that democratizes the diagnosis of Alzheimer's disease.
Having an objective marker that could really improve the accuracy and at a minimum give you a rule out Alzheimer's. So if you're a negative, then they would move you down to see if you have something else. And in a true ultimate fashion, be a rule in, and that if you are Lympro positive, you're certain that you have Alzheimer's. So that's a major fundamental change to clinical practice.
The Lympro news was shared by Amaranthus at the AAIC conference held in Washington last July. While the results seem positive, many scientists believed it is too soon to tell.
Well, they were successful at sorting out Alzheimer's and healthy controls. They found that 76% sensitivity for diagnosing Alzheimer's and a 70% specificity. In well-worked-up patients, the actual diagnostic accuracy is closer to 85%, but the other fault is they did not have yet any of the other biomarkers on these patients to actually support that they truly do have Alzheimer's disease and not something else. The other issue is that they, of course, are just in the first stages, and the next step is to compare patients with other neurological conditions and see whether there, in fact, is a way to distinguish these other neurological conditions based on an immune response of the white blood cells. And I think that's going to be considerably harder to show than compared to healthy controls.
A new type of passive immunization is being tested right now, and it seems to decrease Alzheimer's progression in the earliest stages of the disease. Kaya, can you tell us more about the Solanezumab drug and what it could represent for AD patients in the future?
At the last Alzheimer's Association International Conference, the pharmaceutical firm Eli Lilly shared interesting results from its still ongoing Solanezumab testing. 1,000 patients were tested in what Eli Lilly called Expedition 1 and Expedition 2. During 18 months, half of them were given active treatment and the other half were given a placebo. No significant difference was noted at that time between the two groups. However, when they started to dissect the results, they noticed that the mild AD subgroup showed significantly less decline than placebo-treated patients. All patients were offered to continue to an open-label phase for another 2 years. Secondary results were quite impressive, showing a 34% decrease in the cognitive decline speed of early-stage Alzheimer's patients.
So what we found is that there was a separation between those two groups during the first 18 months, where some were on placebo and some were on active treatment. But once everybody goes on active treatment, the lines essentially stay parallel. So there's a difference at the end of the placebo-controlled period, and just as the theory predicted, actually, that difference doesn't change then during the subsequent 2 years when both groups are being treated with the Solanezumab.
Well, these results are promising. So what's the next step?
Expedition 3 is ongoing, and the last patient's visits are scheduled for October 2016. We should get more results by next year.
It really is a hopeful time. Again, these need to be confirmed. They're preliminary, but so far the results look like Solanezumab could be the first disease-modifying treatment for Alzheimer's disease. So I think we're all just keeping our fingers crossed. We're waiting for the Expedition 3 results next year, and we hope that that will confirm what we've seen so far.
So what did the scientific community have to say about this study?
Reactions were generally enthusiastic at the last AAIC conference in Washington. I spoke earlier with neurologist Dr. Ziad Nasruddin regarding the significance of these results. What do you personally think of the trial's results as an expert on the subject?
In this study, patients who were on active treatment in the placebo-controlled phase kept their treatment benefit at 6 month and 12 month of the open label phase compared to the placebo-treated patients who started the active treatments almost 2 years later. This finding argues well for a possible disease-modifying effect.
According to one critic, the primary outcomes were worse than null, they were negative. What do you think? Should we still have hope for this treatment?
Even if there was a decline over time, the early start group declined 34% less than the placebo group. So I think this is significant.
Can we expect positive results for more advanced stages of Alzheimer's or only the early stages?
At the moment, early intervention seemed to offer a better chance of curbing cognitive and functional decline, but symptomatic-oriented treatments will probably be useful for later stages of the disease.
Thank you for your time, Dr. Nasruddin.
Thank you.
A new study led by Dr. Fernandez of the University of Montreal's Research Center found that deposits of fat in the brain could be a trigger for Alzheimer's disease. This study was conducted to understand why the neural stem cells, which normally repair brain damage, are not working properly in Alzheimer's disease. When compared to healthy brains, significantly higher amount of fat droplets were found post-mortem in the brains of 80 patients. Researchers found similar fatty acid deposits in mice that were genetically modified to develop Alzheimer's disease. It was found that these fatty acid deposits are responsible for suppressing the regenerative functions of the neural stem cell activity. Between 2001 and 2012, Dr. Tuzinski from the University of California conducted the first human clinical trial in gene therapy on early-stage AD patients. 10 patients were administered a single dose of nerve growth factor gene, and their brains were examined post-mortem. Brain examination showed promising results. Cellular growth was identified through axonal sprouting in all patients, and some even showed neuronal hypertrophy, an activation of cellular signaling and functional markers. Since the study showed no adverse pathological effect due to treatment, further clinical trials can be done because the safety issue has been cleared. Dr. Yu from the University of California's Department of Neurology concluded recently the most complete systematic review and meta-analysis on modifiable risk factors for Alzheimer's disease. The analyses included 323 articles with 93 risk factors spanning a population pool of more than 5,000. Very high levels of the amino acid homocysteine in the blood, as well as depression, were found to significantly increase the risk of developing Alzheimer's. The following pre-existing conditions were also found to increase the risk of developing AD: frailty, carotid arteriosclerosis, hypertension, low diastolic blood pressure, type 2 diabetes. It also appears from this study that lifestyle factors with the most influence on the risk of developing AD are low education level and high or low BMI in midlife. This paper also identified some protective factors: the use of estrogen, statin, antihypertensive medications, non-steroidal anti-inflammatory drugs, folate, vitamin E and C, and coffee. These can all potentially reduce the risk of developing AD.
As of today, the only way to ascertain that a person has Alzheimer's is to perform an autopsy of the brain that shows the presence of amyloid plaques and neurofibrillary tangles. Up until recently, physicians have based their diagnosis of AD on clinical observations, using standardized tests and clinical criterions. However, these clinical methods, while efficient in determining whether or not a disease is present, are lacking in their ability to differentiate between normal aging, mild cognitive impairment, and the different stages of Alzheimer's disease. Thus, there's a growing need for biomarkers that are sensitive to subtle brain changes occurring prior to the onset of clinical symptoms, when the potential for preserving various functions is at its greatest. In vivo brain imaging, like positron emission tomography, or PET scan, is a promising biomarker for the early detection of AD. A PET scan is obtained through a nuclear scanner, and it allows one to visualize abnormalities in brain structure, function, and histopathology by using different types of tracer molecules. When the biologically active molecule chosen for PET scan is fluorodeoxyglucose, a molecule in the glucose family, it is called an FDG PET. Brain pathologies such as AD greatly decrease brain metabolism of both glucose and oxygen in tandem. FDG PET measures regional glucose use in the brain to help differentiate Alzheimer's disease from other forms of dementia, and also to make early diagnoses of Alzheimer's disease. The FDG PET scan is the most common type of PET scan. Other types of molecules, known as amyloid beta tracers, can also be used to perform what we call an amyloid PET scan. As you know, Alzheimer's disease is associated with brain lesions caused by a buildup of beta amyloid plaques. Amyloid beta tracers will bind to those amyloid plaques and be reflected on the scan. This type of scan is highly sensitive to the presence of plaques, but a positive amyloid PET scan does not establish a diagnosis of AD or any other cognitive disorder. It's not very specific as to the type of disease we are dealing with. However, a negative scan indicating sparse to no amyloid plaques is inconsistent with a diagnosis of AD, guiding us towards another diagnosis. In an ideal world, combining both FDG PET scans and the amyloid PET scans would provide us with more accurate diagnoses. But PET scanners are expensive to operate and not widely available. In Canada, for example, less than 14 scanners are available per million inhabitants. And as the medical radioisotopes tracers used with PET scan undergo a fast radioactive decay, they must often be shipped to PET scanning centers by air transport, adding to the cost. Being in contact with the extracellular space of the brain, cerebrospinal fluid contains biomarkers that can provide information about the changes in the brain. The cerebrospinal fluid is extracted by a lumbar puncture, using a needle in the patient's back near the spinal column. The three cerebrospinal fluid biomarkers that have shown potential diagnostic reference are amyloid beta 42, total tau, called t-tau, and phosphorylated tau, called p-tau. Low levels of amyloid beta 42 reflect Alzheimer's disease metabolism and the formation of amyloid plaques in the brain. Elevated levels of t-tau reflect axonal damage and neuronal degeneration. Studies have shown a threefold increase of t-tau in the cerebrospinal fluid of AD patients. The combination of t-tau and amyloid beta 42 discriminates very well between brains with AD and normal healthy brains with a sensitivity of 85% and a specificity of 86%. P-tau reflects the brain-tau phosphorylation and neurofibrillary tangles formation. A threefold increase in p-tau has also been shown in AD patients. Elevated levels of t-tau and p-tau in the cerebrospinal fluid reflect degenerative processes in the cortical regions typically affected by Alzheimer's disease. Structural imaging based on magnetic resonance, known as MRI, is an integral part of the clinical assessment of patients with suspected Alzheimer's dementia. MRI is well placed to contribute to an earlier, more precise diagnosis and measurement of progression. Scoring the atrophy of medial temporal structures in the brain using scaling systems such as the Shelton scale is increasingly considered to be a valid diagnostic marker at the mild cognitive impairment stage. Rates of whole brain and hippocampal atrophy, as measured using MRIs, are sensitive and powerful markers of the progression of neurodegeneration. Structural imaging is also included in diagnostic criteria for the most prevalent non-Alzheimer's dementias, reflecting its value in differential diagnoses. The utility of MRI structural imaging will be increased over time by the development of standards and robust algorithms for automated assessment. It certainly shows promise for an easier diagnosis of AD in the future. Blood is a very complex fluid. It contains proteins, peptides, lipids, and metabolites that monitor the physiological activity of the brain. The plasma biomarkers are a very promising diagnostic field for AD because they are less expensive and more accessible than the other diagnostic tools we've discussed so far. You only need a peripheral blood sampling. In the years to come, we'll see more and more research done in the particular field of biomarkers. The goal is to find an easily measurable biomarker, or a combination of biomarkers, that would be very sensitive and very specific for an early detection of AD before even the first symptoms appear. Until then, Alzheimer.tv will keep you in touch with the latest developments in biomarkers.
Thank you for watching this second episode of the Alzheimer.tv News Bulletin. We'd like to hear from you, so if you have any comments or would like to contact our team, please visit the Alzheimer.tv website.